So just to start off, we're obviously here to talk about the phase 1 trial. So I was wondering if you could just give me a rundown of the trial design itself, and then any primary and secondary endpoints.
Sure. Focus C9 is the name of the study. It builds upon some of our learnings from our earlier development programs in Huntington's disease, and it deploys a new type of oligonucleotide backbone modification, a chemistry, I should say, or a molecule that employs a new type of technology that appears to increase the amount of oligonucleotide that can get into tissues and engage targets, so more potency, and also be more durable. So the possibility of single doses leading to durable effects is something that has what we've seen in our animal studies. So on that foundation, the Focus C9 study is targeting C9-associated ALS and FTD. So these are patients who have a hexanucleotide repeat expansion that leads to several secondary effects that ultimately lead to neurodegeneration. These effects are, first of all, an insufficiency of normal C9 or 72 protein, which is important in neuronal function and immune function. In addition, the expansion of the mRNA transcripts leads to the production of these very long RNAs, which in and of themselves are toxic. So you have the toxicity of these extra long mRNAs, and these mRNAs are also translated into toxic proteins, what are called dipeptide repeat proteins. And these proteins themselves are also toxic. And in fact, there's a sense translation and an antisense translation that leads to, again, these toxic proteins, which in and of themselves are toxic. So in this form of ALS and frontotemporal dementia, you have this haploinsufficiency, you have the toxic RNA, you have the toxic protein. All of this through some mechanism leads to gradual neuronal cell loss and death. So our approach is to try to target the disease at that transcript origin. So try and target that extended transcript, and try to do it in a way that brings down all the toxic effects, so the toxic RNA and the toxic protein, without impacting the normal C9orf72 protein. So you leave the beneficial effects intact. And you can do this by targeting different variants of the transcript, because there's several variants produced during transcription. One of those produces toxic effects, the others produce normal effects. So we target the one that leads to toxic effects. And the approach would be to bring down those toxic transcripts, bring down the toxic protein, leave the normal protein intact. So the way we've set up the study, and you do this with an oligonucleotide, and you administer this oligonucleotide, a short strand of DNA RNA that is modified at the backbone level to be a little bit more stable and to have other features, including this new PN chemistry, it's called, that helps in its potency and its durability. And we do that by introducing it intrathecally, get it into the CSF, and then distribute throughout the central nervous system, engaging these transcripts, which are in the brain and spinal cord. So the Focus C9 study is designed to assess the impact of this intrathecal administered oligonucleotide on these proteins, the dipeptide repeat, one in particular called poly-GP. And the idea is, if we can engage that, if we hit the target, if we hit those toxic RNAs, we're going to lower the toxic proteins, and that's actually something we can measure in the CSF. So the study is designed where we're asking for patients who have C9-associated ALS or C9-associated FTD with this expansion, and we're treating them intrathecally and doing 4 cohorts of escalating doses. that we administer a dose to a group of patients. We we do lumbar puncture several times around each of the doses. And as part of those measurements, we're able to see whether the drug is affecting this poly-GP protein. And then the data go to a safety committee or a data safety monitoring board that reviews the data. And then they come back to us and say, okay, increase the dose level this much, change the dose frequency this much. That allows us to then continue to dose a next group of patients. So that's called an adaptive study. So we're doing an adaptive study, and we're also doing a study, what's called a basket-like study, because we're studying ALS and FTD patients at the same time. So through this adaptive design, where we're dose escalating and changing the dose frequency based upon an independent committee's review of the data for FTD and ALS patients, the objective is, again, to see, are we impacting these toxic proteins? Are we impacting other biomarkers like neurofilament light chain, which is a marker of axonal injury? And are we affecting other measures of disease activity, clinical, imaging, et cetera? And depending on the results of this study, this would inform our ability to do a registrational or a larger study in each of these populations, ALS and FTD, which would hopefully result in the measurement of positive effects in the clinical endpoints, and thus allow it to become a treatment. So the study is designed to be adaptive, is designed to change depending on the data, and hopefully accelerate as quickly as possible, because these are two devastating diseases with a lot of impact on patients and their families. So the study, again, is going to be driven sort of like how studies are done in oncology, because the oncologists have figured out a way, let's figure out if these drugs have an effect on the underlying pathology. And if so, let's move them through the process as quickly as possible. Based upon how we view these diseases just as devastating as cancer, so we try to apply this approach to these diseases similar to our colleagues in oncology, and we're hopeful that we'll see a benefit.